So, dear students, so today our topic is catabolism of amino acids. So, before starting our today's sessions, so I should ask one question for you. So, what is the meaning of catabolism? Okay, anyone? Hmm? Today our topic is that the catabolism of amino acids because amino acids cannot be stored in our body. So, when they are enter in our body, when they are formed in body, so they are immediately utilizes. So, they never stored. So they utilize for synthesis of proteins or utilize for synthesis of the biological compounds or they will be excreted as a ammonia. Okay, so that's why the catabolism of amino acids is the most important. It was helpful for the diagnosis of the disease also or how that organ can be work. That is also diagnosis with the help of the to knowing the whatever the end products of the catabolism of amino acids are there. So the secretions in the blood and urine, so that will be the give the clue regarding the what is the scenario of the organs in our body. So that's why the catabolism of amino acids is the most important. It reflects the conversions of that is our metabolism of amino acids into the another form. Okay, so now we can go for the regarding the introductions. So first we can go for the understanding what are the amino acids. So amino acids are the derivatives of carboxylic acid formed by a additions are substitute of the amino functional group at a alpha hydrogen site or alpha carbon atom. So where amino groups is present on the carboxylic acid and such compounds are known as a amino acids. And already you know, so we can got the amino acids from the source of the dietary proteins whatever taken the food, so that food was digested and they liberate the amino acids. And as well as so in our body, so due to various biochemical process or biological process, certain amino acids are synthesized by breakdown of the proteins in the body or body protein. Then also we can got the amino acids. So those liberated amino acids approximately 75% of liberated amino acids, so they are utilized for synthesis of the new protein, so that was required for the on that conditions or on that organs. Okay, so 75%, so whatever the amino acids are liberated, so they are utilized reutilized for synthesis of the new hormones, new proteins, enzymes or development of the organs or formations of plasma membrane. It was required certain proteins or formations of organelles or cell multiplication, so they are required of proteins. So according to that situations, so these amino acids utilized there, then they involved in the synthesis of proteins. And remaining 25% of the amino acids, so they are essential or they are served as precursors for the important biological compounds. Okay, 75%, so they are reutilized, and remaining 25%, so they are involved in the synthesis of the biological compounds because the amino acids cannot be stored immediately. It can utilized or immediately it can be catabolized. So already you know regarding the amino acid pole, that is the processing of the amino acids in our body from the last class. So once again we can review regarding the amino acid turnover or amino acid pool. Okay, the source of amino acids, so in our body is either dietary proteins, so that was digested in the stomach as well as in the small intestine, large intestine, then they are liberated amino acids, so they are enter in our body. So like that, so dietary protein is the main source for the amino acids as well as the body proteins. Okay, so that is also the source for the amino acids. So in our body, especially in the liver, the protein can be synthesized. Okay, so when it required to synthesis of certain hormones or other any substance in any parts of the body, so then liver it can release the protein that was catabolized, Then they releases amino acids. So those amino acids they travel in the blood, then they reaches to the target site, then were they involved in the to synthesis of the desired proteins. And also these amino acids they are involved in the synthesis of the nitrogen containing compound. They can undergo the catabolic process that was known as the transamination. Then so they are involved in the formations of the alpha ketoglutarate. Then from the alpha ketoglutarate, so they involved in the glutamate. And glutamate it can undergo the deamination. Then it was involved in the formations of ammonia and urea. So like that, the remaining excess of the amino acids, so they undergo the catabolism and excreted from the body. Or some amino acids, so they are used for synthesis of the energy. They can undergo the gluconeogenesis, then they are produces the energy. Or some, so they are required for the productions of glucose, and some they are utilized for the productions of fats or synthesis of fats, and some they are required for the synthesis of the non-essential amino acids. So whatever the dietary amino acids are there, so they are utilized for synthesis of the non-essential amino acids. So in our body, so like that, so the amino acids, so they are processes in our body, either the formations of waste material or utilized purpose of synthesis of proteins or energy purpose or glucose fat synthesis purpose or synthesis of the non-essential amino acids. So now we can learn regarding the need of amino acid metabolism. So it is utilized to generate the energy. Okay, so excess of amino acids, so they can undergo the gluconeogenesis or amino acid metabolism. So then, so they are utilized the they are generate the energy. So that energy was helpful for the processing or regulations of the biochemical process in our body. So as well as, so that amino acids is helpful for synthesis of the glucose, and also so they are helpful for the formations of fats and ketone bodies. And ketone bodies fats are the bioenergetics compound or energy rich compounds. So they are formed from the amino acid in the case of starvations or in the case of the deficiency of ATP. So then these ketone bodies and fat they are utilized for the purpose of the synthesis of the ATP. So that's why the amino acids they are play an important role continuously to supply the energy to the body by involving in the formations of fat and as well as the formations of the ketone bodies. And also, so they are involved in the productions of the non-essential amino acids. Okay, certain there are the total 11 amino acids are the non-essential. So they are synthesized from the excess of the amino acids or dietary amino acids. So like that, the amino acid metabolism it is a mandatory or continuously regulated in our body. The purpose of energy, purpose of synthesis of energy compound, and the purpose of synthesis of the non-essential compounds. So next one, so regarding the catabolism or metabolism. Okay, so regarding catabolisms or metabolism of amino acids, so it is a breakdown of amino acids. So amino acids are mainly metabolized or catabolized by four process or in the four mechanisms. One is the transamination, that is the transfer of amino group from one amino acid to the alpha keto acids. Okay, then it was responsible for formations of the alpha keto acid. From the alpha keto acid it was involved in the formations of the glutamate. Okay, so that reaction, so where the amino acid can be transported from amino group, okay, from the amino acid is transported to the alpha keto acid. So that reaction was known as the transaminations. And another is a mechanism that was involved in the catabolism of amino acid is a deamination. Deaminations means removal of the amino group from the amino acid. Okay, transaminase means the transfer of amino group from amino acid to the alpha keto acids. And regarding deamination, that is the removal of the amino group from the amino acid. Then that amino group is enter into the metabolism or various reactions, then it can forms the ammonia then into urea. So like that, that was excreted, and such reactions was known as the deaminations. Once again, the deaminations are two types: oxidative deaminations and direct deaminations or non-oxidative deaminations. So these are the enzymes, so they are involved in the oxidative deaminase like glutamate dehydrogenase in mitochondria, amino acid oxidase in the peroxisomes. And regarding the direct deamination, non-oxidative, that is deaminations of dehydration, the removal of water, or deaminations by a desulfidations, the removal of the H2S. So these are the example, or these are the enzymes, those are responsible for the formations of the ammonia or transfer of the or removal of the amino group from the amino acid. Such reactions was known as the deaminations, and both transamination, deamination, they are occurs simultaneously or at a time. Okay, so next one is the decarboxylation, that is the removal of the carbon dioxide from carboxylic group of amino acid, and such reactions are known as the decarboxylation reactions, where the carbon dioxide was removed from the carboxylic acid from the amino acids. Okay, then so they yield the amines, and those amines are the physiologically and biologically more active. So next one regarding the transmethylation, that is the transfer of methyl group from one to another compound or another amino acid, and such reactions or catabolic reactions of amino acids are known as a transmethylation. So like that, so there are the four mechanisms and four reactions. So they are involved in the catabolism and as well as the metabolism of amino acids, like a transaminations, deamination, then decarboxylations, and as well as the transmethylation. So now we can learn one by one. So first we can go for the transamination. Simply, the transaminations mean the transfer of the amino group from an amino acid to a keto acid. That is the transfer of, that is the amino group from amino acid to the keto acid. So that catabolism or that reaction is known as a transamination. And transamination is mainly occurs by a enzyme is known as the aminotransferase by a one settle that was known as the PLP, that is the pyridoxal phosphate. So it is the complex. Okay, it consists of the pyridine phosphate from the vitamin B6. So this pyridoxine phosphate or PLP, so along with this enzyme, so that was helpful for the transfer of the amino group, because so pyridoxine phosphate, it can accept the amino group first in the presence of aminotransferase enzyme. Then so that amino group transport to the keto acid, then the that is the aminotransferase, that then the enzyme was separated. So like that, so this enzyme, that is the transaminase, it was helpful for the interconversion of amino acid into a keto acid and keto acid into a amino acid by catalyzing the reactions or by transporting the amino group. So in the presence of the pyridoxal phosphate, it was obtained from the pyridoxine or vitamin B6. Okay, so this PLP, that is the pyridoxal phosphate, it was play an important role to occupy the amino acids first, then it's applied to the keto acids in the presence of the aminotransferase enzyme. So like that, the transamination reactions or the transfer of amino group from amino acid to the keto acid, so that was occurs in the presence of the transaminase enzyme, that was also known as the aminotransferase enzyme. So this enzyme is mainly active in the both cytoplasma and as well as the mitochondria. So this enzyme, so along with the PLP, it was helpful for the transfer the amino group from amino acid to the keto acid. Then it can form the new keto acid. That is the R1 is the one amino acids. It indicates. Then here, so same amino acid it become the new keto acids. Then this keto acid it become the new amino acid. So like that, the interchange was occurs by the presence of aminotransferase, and this enzyme is active in both cytoplasma as well as the mitochondria. So examples are group of the transferase enzymes are that is the aspartate aminotransferase. It is the example of transaminase enzyme. Or alanine aminotransferase, that is ALT. Then as well as glutamate oxaloacetate transaminase, it is also written as a GOT. And glutamate pyruvate transaminase, it is also known as the GPT. So these are the many enzymes are there. So among them, so these are the four main enzymes. So they are commonly involved in the transaminations reactions. Among these four, so these two, that is the AST and ALT, frequently involved in the catabolism of amino acids. Okay, and the catabolism of amino acids, it was responsible for to cause of the negative nitrogen balance. Okay, so already you learned from the last class that is the catabolism is more. Okay, then it indicates that the outcome is output is more than as compared to input. So that was known as the catabolic effect. And catabolic effect, it was indications of the, that is the various disorder like starvations and acute and chronic illnesses, and as well as the protein deficiency. And these enzymes are mainly involved in the transamination or catabolism of the amino acids. And estimations of these enzymes from the blood or from the plasma, so that was helpful for the diagnosis of the various disorders. If you have any symptoms related to the that organ, particular organ, then you can go for the plasma or blood estimations of these enzymes. If this enzyme is found and particular pain in that part of the organs or any changes in the physiological appearance in that organ, and as well as, so these enzyme if they are found more in our blood, means that indicates so that organ is under the malfunction, it was suffering from the various kind of the disorder. So like that, so the estimations of these enzyme aminotransferase enzyme, so they are helpful for the diagnosis of the pathological variations in the various organs like a kidney, liver, then as well as gallbladders or many other, even heart, brain, because so these enzymes frequently found in the those organs. And estimations of these enzyme only they are, these are found in our blood in the more concentration only when so that organ under the inflammations or that organ under the malfunction, so then only the catabolism is occurs. So there is no anabolic reaction is present due to malfunctions of that organ. And these enzymes, because amino acids cannot be stored, either it is reutilized, either it is metabolized, so that cannot be stored. If it is not anabolized means it can be catabolized. If it is catabolized means, so these enzymes are more secreted, more activated, more present, then they are more secreted into the blood, in the blood, so they are more number is present. As the concentrations of these enzyme so in the blood is increasing, means it can denote the some organ so that was under the malfunction. So later on the doctor they can go for the diagnosis of ultra scan or scanner or CT scan. They use the CT scan for diagnosis of the various organs. So whether which organ will be the malfunction, because that enzyme was the found in the blood. So that's why the transaminations that was occurs in the presence of transaminases, and that the diagnosis of transaminase or estimations of transaminase, so that was helpful for the diagnosis of the various disorders. So regarding all transamination reactions, alpha ketoglutarate, that is a alpha kz, acts as a amino group acceptor. Okay, that acts as the amino group acceptor. So first, so this enzyme along with this PLP, it carries occupy this enzyme and finally supplied to the alpha, that is a ketoglutarate. So that's why the alpha ketoglutarate in the all the transformation reactions, it is acts as the amino group acceptor and involved in the formations of the new amino acids. Okay, or it can convert amino acid into keto acid. Then most all amino acids undergo the transamination reaction except lysine, threonine and amino acids. So they do not undergo the transamination reaction. But maximum amino acids, so they are catabolized or they are metabolized by a transamination reactions that accept the lysine, threonine and as well as the amino acid. So now you can go for regarding the characteristics of the amino that is the transamination reactions. Okay, transamination reaction is the one of the main reaction where the amino group can be transported from amino acid to the keto acids, and it is the one of the catabolic reactions or metabolism of the amino acid. That was the transaminations. So all transamination reactions are all transaminase enzymes. So they require the pyridoxal phosphate as a coenzyme, and pyridoxal phosphate is the source of the vitamin B6. It means the B6 vitamin, that is the B complex vitamin, that was play an important role in the transaminations reactions. So dear students, the transamination reaction is also one of the important reaction, so that was helpful for continuously to supply the amino groups for synthesis of new amino acids and new proteins, and as well as the transamination reaction is essential for synthesis of the non-essential amino acid that was required for the synthesis of hormones, synthesis of new proteins, synthesis of enzymes, synthesis of energetic compound. So they require certain non-essential amino acids also, and source of non-essential amino acid is that is the transaminations reactions. So that's why the transamination reaction is helpful for catabolism of amino acids as well as the synthesis of the new non-essential amino acids. And what are the enzymes? So those are involved in the transaminations reaction. So they are mainly depending upon the presence of coenzymes like a pyridoxal phosphate. And pyridoxal phosphate source is a vitamin B6, and vitamin B6 cannot be synthesized in our body, that was got from the food. So that's why if you need to develop your body to maintain the growth, continuous growth in your body means you should be give to your body regarding the vitamin B6. It continues to supply the pyridoxal phosphate and continuously transformation reactions occurs, then it can give the new amino group and new non-essential amino acids, and those are involved in synthesis of the new proteins. So then it is the another side. Then another characteristics of the transamination reaction is in the specific transfer exist in each pair of amino acids as well as keto acids. However, only two, namely aspartate transaminase, alanine transfer, make a significant contributions for transaminations. Okay, so in the transaminases, so they are only so you involved in the utilize two pair of amino acids, that is the one the amino acids and keto acids were only, so they can interchange the amino groups, and in which so these only two enzymes, so they are significantly contributions in the transaminase like aspartate transaminase and alanine transaminase, that is AST, and estimations of these enzymes in our blood, that was helpful for significantly helpful for the diagnosis of the conditions of the organs. And next one, so transamination, so there is no liberations of ammonia as ammonia is taken plus. During transaminations, amino acids are catabolized because ammonia is the end product or metabolism of the amino acids. But transamination is also metabolic reactions or catabolic reactions of amino acids. But in transamination, the ammonia is cannot be liberated. Only the transfer of amino groups can be occurs. It is another characteristics of the transaminations reactions. And all transaminations reactions are reversible in both side. So those reactions are regulated, so they transport the that is a amino group to the keto acid, and later on if their need means that particular amino acid, they can reverse take the amino groups, then involve the formations of that particular amino acid. So that's why so all the transaminations reactions or what are the amino acids so they are involved in the transformation reactions, so they are reversibly reformed. So that's why the transaminations reactions is a reversible reactions. And transamination is very important for redistributions of amino groups. Transamination is helpful for the transfer of the amino groups or redistributions of amino group, or continuously they supply the amino groups for productions of the certain non-essential amino acids. So like that, so those are non-essential amino acids are there is no need to take from the dietary source, because due to transformation reactions, so that was present in our body, so they are helpful for the, or they supply the amino group and helpful for the productions of the non-essential amino acids. Another feature characteristics is a transamination diverts the excess of amino acids toward the energy generations. So same reaction, it is also put to the amino acids, there is excess of amino acids that was left by formations of biological compound as well as the formations of protein, then they transports towards the generations of energy by undergoing the catabolic reactions, gluconeogenesis, many others reactions. That is one of the another characteristics of the transaminations reactions. So next one, the amino acids undergo the transaminations finally, so they can concentrate the nitrogen in a glutamate. So nitrogen, it was play an important role in the synthesis of proteins as well as synthesis of the amino acids. So these transamination, so they can continuously supply the nitrogen or deposit the nitrogen in a glutamate. So that's why the glutamate is an the amino acid, only one amino acid. It can undergoes the oxidative deamination and liberate the ammonia. That ammonia is required for the synthesis of urea. Okay, in all the transamination reactions, the end product is a glutamate, and glutamate is the, it is the only one amino acids. It can undergo the oxidative deamination and it can liberate the ammonia for synthesis of the urea. It means the urea that was synthesized from the ammonia, and ammonia was synthesized by the oxidative deaminations of the glutamate. And glutamate is the product of the all the amino acids transamination. So they are count after transamination, so they are counted into glutamate, and also they can load the nitrogen in the glutamate only. Okay, next two characteristics, the transamination is not restricted to alpha amino group only. Okay, it can occurs in the all the sites. Okay, it is not restricted to the only for alpha amino group or those amino group, so they are attached to the alpha carbon atoms. So it is also happens where the amino groups are present, so where it can be removed to from that amino acid and supplied to the keto acid. So that's why it is not restricted to a particular alpha amino groups only. Then serum transaminases are important for diagnosis or prognostic purposes. So if you estimated serum transaminases, so they are helpful for the diagnosis and as well as prognostic. Prognostic means how the disease can be cured. Okay, if the liver is problem is there, so on that time you can estimate the enzyme level, then you can start the therapy, and month by month you can estimate the enzyme. If the enzyme level day by day or week by week or month by month that was decreases, means it can gives the idea regarding the progress of the disease. So that was known as the prognostic purposes. So this was helpful for to note down the whether the patients or persons or cure from that disease or therapy is sufficiently cure the disease or not. That is also diagnoses are estimated. So with the help of the estimations of the transaminases from the our blood. So these are the various characteristics of the transaminations reaction. So now we can go for the regarding the mechanism of the transaminations reactions. So all aminotransferase, so these are the enzymes that was involved in the transamination reaction. So they are required the pyridoxal phosphate, that is the PLP, which was derived from the ones that is the vitamin B6. So transamination reactions, so are aminotransferase enzymes. So they are not active in the absence of the coenzyme or cofactor like a pyridoxal phosphate. It is also written as the PLP. And transamination reactions, it was mainly occurs in the two steps. In the first steps, that was involvement of pyridoxal phosphate in the transfer of amino group. Okay, that is the amino group of amino acid is transferred to the pyridoxal phosphate, then forming the pyridoxal phosphate. Then it can release the keto acid. Okay, that was seen in the, so these first reactions. Okay, so that pyridoxal phosphate, it can accept the amino group from the amino acids. Okay, then this pyridoxal phosphate, it can conjugate with the transferase enzyme. Okay, so this pyridoxal phosphate, it can conjugate with the that aminotransferase enzyme. Then it can accept the amino group from the alanine and involved in the formations of pyruvate and alpha, that is alpha ketoglutarate. And later on, so this enzyme was separated. Okay, then the from the alpha ketoglutarate, it was converted into a glutamate. That is the second step. So in the second step, the alpha ketoglutarate, it can react with the pyridoxine phosphate, forming a glutamate. So in all the reactions, in all the transformation reactions, the glutamate is the end products, and this glutamate only, it can undergo the deaminations reactions. Then it was involved in the formations of the ammonia and as well as urea. And this step is also reverse. In the reverse also, the same enzyme is involved that can convert glutamate into alpha ketoglutarate, then into presence of the pyridoxine phosphate, then it was converted into alanine. So there is the only two steps. So they are involved in the transamination. One is the amino group can of the amino acid is transported to the pyridoxal phosphate, forming the pyridoxamine phosphate and releasing a keto acid. Then in the second step, the alpha ketoglutarate, it was reacts with the pyridoxine phosphate, forming a glutamate. So this is the mechanism of the transamination. And between these two reactions, another additional steps was involved. It was the hidden steps regarding the, so that enzyme that was known as the formations of, that is the enzyme PLC skip base and amino acid PLP skip base. That is the amino group of the pyridoxal phosphate is then transported to the keto acid to produce a new amino groups. Then that enzyme PLP is generated. Okay, so this enzyme, it can occupy the that is the presence of the enzyme, and this potassium phosphate, it can occupy the amino group. Then it can transport the amino group. Then enzyme was separated. Then the transamination reaction was finished. So like that, another hidden steps. So it was present in the transamination reactions along with the step 1 and step 2 reactions. So next, we can go for the biological significance of the transaminations reaction. So now we learn regarding the biological significance of transaminations. So that is the transamination used both for the catabolic as well as anabolic process. The breakdown of proteins or amino acid as well as the synthesis of the amino acid. So that's why the transamination, it can utilize in the both catabolic as well as the anabolic process. And second one is the resultant of the alpha keto acids can be completely oxidized, and it can provide the energy, glucose, fats, ketone bodies depending upon the cellular requirements. So whatever the keto acids, alpha keto acids, so that was formed. So that alpha keto acids or alpha ketoglutarate enter in a TCA cycle. Okay, alpha ketoglutarate, so that was enter in the TCA cycle. Then it was involved in the synthesis of the new amino acids or synthesis of glucose or fats and as well as ketone bodies and as well as the synthesis of the ATP. So whatever that is the another significance of the transformation reaction. So that was helpful for the productions of energy, synthesis of glucose, then synthesis of fats or ketone bodies. Then since it is a reversible process, it is also used for the synthesis of the non-essential amino acids. And transaminations, it was helpful for to equilibrating the amino groups, or it can continuously supply the or provide the amino groups. It acts as the source of amino acids from the available from the alpha keto acids. That is the example is a from the alpha keto acid it was converted into glutamate, and glutamate it has a serve as the providers for the amino groups that was required for the various process or required for the synthesis of the new amino acids. And in our food, that the dietary amino acid it is a rich of the glutamate. Glutamate is also it richly presents in our dietary proteins as a amino acids. So that was undergo the deamination. It can produce the amino groups, or also it is involved the formations of the urea or synthesis. And what are enzymes? So those are involved in the transaminations reactions, and those are helpful for the diagnosis of the various disorders like SGOT, SGPT, then ALT and AST. Among SGPT, SGOT, ALT, AST, it is the more frequently it was seen in the various disorders of the organs. And estimations of these enzymes that was helpful for it can give the clue regarding the progress of disease and therapy, and as well as helpful for the diagnosis of the abnormal physiology and disorders. Okay, so next one we can go for the regarding the deamination. The deamination nothing but simply removal of the amino group from the amino acid as a NH3 is known as a deamination. That is the removal of the amino group from a amino acids. And transamination means transfer of amino group, and deamination means the removal of amino groups. Okay, deamination is the result in the liberations of the ammonia. So that was utilized for the formations of the urea. So whatever the ammonia, so that was liberated during the deamination, so that was utilized for synthesis of the urea. Okay, so the deamination, so they are either oxidative or non-oxidative, so they are regulated by the presence of oxidation as well as the in the absence of oxidation. So this the deamination reaction is occurs, those are occurs are active in the presence of the oxygen means, and those are known as the oxidative deamination, and those reactions occurs in the absence of the oxygen means, and such reactions are known as the non-oxidative deamination. So like that the deaminations reactions, so they are mainly classified into two types: oxidative deaminations and direct deamination, that is the non-oxidative deaminations. And oxidative deaminations, the important enzyme is a glutamate dehydrogenase. It was found in the mitochondria. Then another is a amino acid oxidase. It was found in the peroxisomes. It means the deaminations reaction it was occurs in the both mitochondria as well as the in the cytoplasma in the peroxisomes. So in the presence of the glutamate dehydrogenase enzyme, so as well as the amino acid oxidase enzyme. So regarding the non-oxidative deaminations, the example is the deaminations by dehydration, that is removal of water, or deaminations by desulfhydration, that is the removal of the H2S. So these are the example for the non-oxidative. So they are occurs in the absence of the oxygens. And here one notice there. So both transamination and deamination they occur simultaneously at a time in the same site. So they are occurs same time, the transamination is occurs, and same time the deamination is occurs. So that's why these reactions are known as the are such reactions, so where the transaminations and deaminations is occurs simultaneously, and such reactions are known as the trans deamination. That is the trans deamination. It indicates the both reactions, so they occurs simultaneously. Okay, so that's why, so these are known as the trans deaminations reactions. So next one, so regarding the trans deaminations, so this is the first step. So here it is the transamination is occurs in simultaneously. So what is the glutamate is formed, so that glutamate it was enter into the deaminations reaction, that is oxidative deaminations reaction. So that's why the both transamination and the deaminations reactions occurs simultaneously. So the transaminations in the presence of aminotransferase and PLP, alpha keto is converted into ketoglutarate is converted into glutamate. Okay, then so this glutamate is enter into the that is a deamination oxidative in the presence of glutamate dehydrogenase enzyme and utilize the coenzyme like NADP or NAD. Then it was involved in the formations of the ammonia. Then it can enter into the formations of the alpha keto glutarate. So like that. So that's why. So these both reactions occur simultaneously. So that's why these are known as the transaminations, where the transaminations and deaminations occur simultaneously. In the transamination occurs in the aminotransferase, the end product is the glutamate. And the deamination, so that glutamate is entering into the deaminations. Then it was involved in the formations of the ammonia in the presence of the GDH, that is the glutamate dehydrogenase, and as well as cofactor is the NAD or NADP. So now we can go for regarding the oxidative deamination. So oxidative deamination is the liberation of free ammonia from the amino group of amino acids coupled with the oxidation. Simply, that is the oxidative deamination means the removal of the amino group from the amino acids in the presence of the oxygen or by a oxidations reactions. And the oxidative deaminations, it can occurs or takes place in both liver as well as in the kidney. So that is the oxidative deamination. It was occurs in the liver and kidney. So the purpose of the oxidative deamination is to provide the ammonia for urea synthesis as well as the alpha keto acids for variety of the formations of the various intermediate for the TCA cycle. Then the energy was the generated. So then the regarding the, so there is a glutamate dehydrogenase. So this is also known as the GDH. So this is the main enzyme. So that was regulate the, that is the oxidative deaminations reactions. So this is an enzyme consists of identical 6 units and molecular weight of the glutamate dehydrogenase is a 56,000 and GDH is controlled by allosteric regulations. Okay, so GDH, it is a enzyme, it is a complex enzyme. It was consist of the 6 identical unit, and it is the regarding it consists of the 50, that is the molecular weight around that is the 56,000. So this is the enzyme. So that was responsible for to convert L-glutamate into amino alpha amino glutamate, and the glutamate dehydrogenase enzyme that was responsible for the conversions of L-glutamate into alpha ketoglutarate, and you can utilize the NAD or NADP as a coenzyme. Okay, from the glutamate, the alpha ketoglutarate is formed. Okay, through a formations of the intermediate compound like a alpha amino glutarate. Then it can liberate the water in the presence of the same enzyme, that is the alpha, that is the sorry glutamate dehydrogenase. Then it can respond to the formations of the alpha ketoglutarate and ammonia. So this ammonia, it utilized for the synthesis of urea, and this alpha ketoglutarate, it can enter in the TCA cycle. Okay, so that's why, so the that is the glutamate dehydrogenase. It was involved in the both anabolic as well as the catabolic reactions. So it was involved in the formations of the alpha ketoglutarate through a formations of the intermediate compound. So like a alpha aminoglutarate by utilizing the coenzymes like a NAD or NADP. Okay, so GDS enzyme, GDH enzyme, so that was controlled by a allosteric regulations. That is the active site. So those are present in the enzyme. If the more active sites present means the more substrate can be bind and more coenzyme can be bound, then the reactions will be accelerated. So that was known as the allosteric regulations and allosteric reactions. Okay, so what are the active sites? So those are binding sites. So those are present in this enzyme. So that can determine the rate of the oxidative deamination. So that was known as the allosteric regulations. And here the GTP, that is the guanine triphosphate, and as well as adenine triphosphate. So they are inhibit the glutamate dehydrogenase enzyme activity. And regarding GDP, that guanine diphosphate and ADP adenine diphosphate, so they are activate the glutamate dehydrogenase. So that's why the GDS enzyme, so that was mainly depending upon the GTP ATP or GDP ADP. If more GTP ATP is energy present in your body, means it was inhibit the deamination reactions. In your body less energy is present, means the GDP ADP will be more. So that's why in the that particular cell, and that why, so this enzyme can be activated, that can convert into alpha ketoglutarate, then it was involved in the TCA cycle, then involved in the productions of the more energy. If the energy was sufficient, then same the ATP can inhibit the, so this is, so like that the ATP GDP, that sorry, GTP ATP and GDP ADP, so they are involved in the activate or deactivate the glutamate dehydrogenase enzyme. Okay, then as well as the steroids and as well as the thyroid hormones, so they are inhibit the GDH, and lack of GDS, the alpha ketoglutarate is not formed, and lack of alpha glutamate, the TCA cycle cannot be regulated. So in the starvations or during the carbohydrate abnormal metabolism, then we do not got the energy if you are using steroids and thyroid hormones in excess. Because if the carbohydrates is not synthesized or not involved in metabolism, means, so on that time the fats or acetyl CoA acetate, they are entering the TCA cycle, then they are involved in the formations of the ATP, that was the blocked due to presence of this enzyme, that is the steroids and hormones. So that's why unnecessarily you do not take the steroidal drugs for treatment of any inflammations or any asthma, any other condition. So regarding the mechanism of oxidative deaminations, so the oxidative deaminations, it was helpful for the deamination or transfer of the amino group from both alpha amino acid or L amino acid and as well as the D amino acid. So this is the oxidative deamination is occurs for both, that is the mm, alpha amino acids as well as the D amino acids, and the enzymes, so that was involved in the oxidative deamination is a, there is L amino acid oxidase for L amino acid and D amino acid oxidase. Okay, that is the D amino oxidase, so that was regulate the deaminations for the D amino acids. And regarding the these enzymes, so this enzyme is less active, but the D amino acid oxidase, it was the more active. Okay, and these both enzyme, that is the L amino acid oxidase and D amino acid oxidase, so these are the flavoproteins. Okay, and these enzymes is acts on the amino acids, there is L amino acid oxidase acts on the alpha amino acids, and D amino acid oxidase acts on the D amino acids, so in the presence of the flavins. Okay, so this, that is regarding the L amino acids, so it is required the flavin FMN, but D amino acid oxidase, it can utilize the, that is the FAD as a coenzyme. That is the, here I write in the note, that is the L amino acid oxidase use the FMN as a coenzyme. So whereas FAD is used as a coenzyme by D amino oxidase, and both enzyme, so these are the flavoproteins, so they are required for the oxidative deaminations of L amino acids as well as the D amino acids, and then so they are formed the, that is the alpha amino acids. From the alpha amino acids, it can liberate the ammonia by hydration reactions. Okay, same enzyme, it can undergo the hydration reactions, then it can liberate the ammonia and forms a alpha keto acids, and then this keto acids, it is enter into a TCA cycle, then it utilized for the formations of ATP, and this flavin, it can utilize the oxygen, or this is utilize the oxygen and form the H2O2, and this H2O2 further it can catabolized by a catalase group enzyme that was responsible for formations of the H2O and oxygen. So like that, so this is the mechanism for oxidative deaminations for L amino acids as well as the D amino acids, and both reactions, so there occurs by a presence of two enzymes, that is L amino acids oxidase for L amino acid and D amino acid oxidase for D amino acid, and both enzyme utilize the coenzyme as a FMN and FAD and involved in the formations of alpha amino acid, and another side involved in the formations of the hydrogen peroxide. Okay, and further alpha amino acid undergo the hydroxylation reactions or various reaction, then forms the ammonia. Okay, then it formed the alpha keto acid, and this H2O2 peroxidase, it was catalyzed by the catalase enzyme and involved the formations of water and oxygen. But regarding the L amino oxidase, so they are less active or low concentrations, but D amino acids, it is a more active, are highly present in both liver as well as in the kidney. This is regarding the mechanism of oxidative deamination. Next one is the non-oxidative deamination, that is the serine, threonine or cysteine and histidine. They can undergo the non-oxidative deaminations and to form a corresponding alpha keto acids. So all the catabolism of one acid, so they are involved in the formations of alpha keto acids. So regarding the deaminations of serine and threonine, so they are carried out by the enzyme dehydratase enzyme, or whereas the uh, deaminations of cysteine and cysteine, it is carried out by the desulfurase, and both enzymes, so they are depending upon the BC. So these are the example, that is the amino acid dehydratase enzyme, it is involved in the non-oxidative deaminations for serine, threonine and homoserine. So these are the hydroxy amino acids. So in the presence of dehydratase enzyme, so they are forms the respective alpha keto acids and release the ammonia. So another enzyme, that is amino acid desulfurhydrase, it is also involved in the non-oxidative deaminations reaction. It can deaminate the amino acids like a cysteine and cysteine. Okay, these are the sulfur amino acids. So in the presence of so disulfuride sulfurhydrase enzyme, they are forms the ammonia as well as the H2S. Then it can involve in the formations of the pyruvate. And third enzyme, so that is the histidase enzyme, that is the deaminations, it is required for the deaminations of histidine. So this histidase enzyme, so that was uh, essential for the deaminations of the histidine and release the ammonia and as well as the urocanate forms. Okay, regarding the steps of the non-oxidative deaminations, so the deaminations was takes place in the two steps. In the first steps, so the amino acid is formed by the actions of the these enzymes, the dehydratase, desulfurase and as well as the histidase. In the second steps, so these amino acids undergo the hydrations and deaminations to produce a alpha keto acid. Then these alpha keto acids enter in the TCA cycle. So this is the mechanism regarding the non-oxidative deaminations. So next one, so regarding the decarboxylation, that is the definition. The decarboxylation of amino acids means the removal of the carbon, okay, the removal of the carbon dioxide from the amino acids with the formations of amines. And what are the amines? So that was formed during the decarboxylation reaction. So they are highly physiologically active. So these are either hormones or neurotransmitters. Okay, whatever the amino acids, so they are undergo the decarboxylation reaction. So they are forms the amines, and those as amines, so they are highly physiologically active. So they are involved in the regulations of the various reactions. And the enzymes, so those are involved in the decarboxylase enzyme, so that enzyme is known as the decarboxylase enzyme, and they are utilized the coenzyme, that was the once again pyridoxal phosphate, and pyridoxal phosphate is obtained from the vitamin B6. So this is the general structures of the decarboxylation. So due to presence of the decarboxylase enzyme, so that amino acids can undergo the decarboxylation reaction, it was involved in the formations of the amines. So regarding the different types of decarbonation of amino acids, so they are undergo the decarbonation at the alpha site. So that's why those are known as the alpha decarboxylations, and some, so they are subjected to the omega site of carbon atom. So those are known as the omega decarboxylation, and some reactions are decarboxylations are occurs with the transamination, and those are known as the decarboxylations with the transamination. And some, so the double amino acids they are involved, that is the decarboxylation with the conjugation with the two molecule, and such are known as decarboxylation with the conjugate two molecules. So like that, the four mechanisms, so they are involved in the decarboxylation of the amino acid. So next one, so regarding the significance of amino acids decarboxylations, that is the, they are involved in the, that are decarboxylations of amino acids involved in the formations of the physiologically active compound. For example, glutamic acid undergo the alpha decarboxylation, then it was involved in the formations of the GABA, and GABA is a neurotransmitter, it is a amine in the CNS. It was helpful for to regulations of the sleep, or it is helpful for to relax the brain, it is helpful for to decrease the were firing capacity of the brain, and GABA is helpful for the treatment or avoid the convulsions or epilepsy. If any problem in the formations of GABA mean, then the persons are suffering from the abnormal sleep or insomnia, or so the person was suffering from the epilepsy. So that's why whatever the amines was found by a decarboxylation amino acids, so they are having the variety of the physiological actions, physiological actions. So another example, that is the histidine, it can undergo the decarboxylation, that it can forms the histamine, and histamine is also amino acids or neurotransmitter. The not amino acid, neurotransmitters are local hormones. So it is involved in the regulations of the blood pressure, are involved in the to regulate the arousals. Okay, we are alert now due to histamine in the brain. Okay, also it is involved in the regulations of memory power. So next one, tyrosine is also tyrosine is amino acid undergo the decarboxylation reaction, that is involved in the formations of tyramine, and tyramine is essential for the formations of the dopamine, formations of the noradrenaline, and formations of the adrenaline. So already know regarding the adrenaline, noradrenaline, dopamine. So they are the sympathomimetic neurotransmitters are involved in the regulations of the various sympathomimetic functions among the increase the heart rate, blood pressures, or many other functions are there. So that's why. So these are tyramine is also physiologically active compound regulate the various biological functions. Another example is a tryptophan undergo decarboxylation reactions and involved in the formations of the serotonin. And serotonin already know it is required for the regulations of the biological clock or circadian rhythm. And last one, that is tyrosine. It is also responsible for the formations of the dopamine through a timer. So like that, the significance of decarboxylation reaction is it is involved in the formations of the physiologically active compounds or amines, and those amines, so they are neurotransmitters or neuromodulators or neurochemicals. So those are involved in the regulations of various functions regarding the sleep, arousal, memory power, muscle contractions, muscle relaxations, as well as heart rate, blood pressures, secretions. These all are regulated by these neurotransmitters or neurochemicals, neuromodulators. So those are produced in our body or in brain specially by decarboxylations reactions by of a amino acid. So this is the important significance of the decarboxylations of the amino acids.